right there. All right. Gotta love Zoom. So now think back to what we talked about. Each amino acid is spaced by 100 degrees. So you're going to count 100 degrees along the spiral. And I'll tell you, these partitions, you can count them yourself, are spaced by 20 degrees. So you just count 20, 40, 60, 80, 100, and you place your next one, which is leucine. Now for your asparagine, you do the same thing, 20, 40, 60, 80, 100, and place it. And you'll just keep on counting. It's a little tedious, I know, but you'll love this type of problem, I swear, until all your amino acids are placed on your helical wheel. So that's pretty straightforward. You just got to remember 100 degrees spaced apart. Now, can someone unmute and let me tell me, is this an amphipathic helix or not? So think about what amino acids you have. It may take a little bit.
I would say it's amphipathic.
Yeah, exactly. And can you tell me where the hydrophobic face is?
So I noticed that there is isoleucine.
Yep, right here.
And there's L.
Yep.
Leucine, yeah.
Leucines.
Which is also in phenylalanine, which is also so like that side you could almost say is the hydrophobic.
Yeah, exactly. So that top side, and here's a line separating the two. So your top side is it's hydrophobic, and you button with a mix. Remember, it'll be probably a mix of charged and uncharged residues. Will be in the bottom. So for this type of problem, yes, it is amphipathic, and for this type of problem, it's really important that you know your amino acids, you know their structures, you know their polarity, because when you're in an exam and you don't have much time, you're going to want to know those instantly. But that's how you do this type of problem. I'm sure Professor Curian will ask you something crazy to do with it, but this is the basic form of it. Does anyone have any questions? All right, then I can hand it off to the next tutor.
Hello, everyone. Can anyone hear me?
Yeah.
Okay. I should be sharing, but I don't see my screen.
We saw a black screen. Yeah, now we can see it.
All right. Can you guys see it? No?
No, I think you stopped screen sharing.
All right. That is unexpected. I'm sorry.
Yeah, now I can see it again.
Cool. Thank you. Yay. All right. Hello, everyone. I am Lisa. Let's talk about Ramachandran diagram today. I know you guys have just been introduced to this topic, so I want to give you some background information to help you think through this concept and maybe talk a little bit about why it will still be useful. Yeah, you should see my face. Okay. So the central idea I want to talk about today can really be captured by the subtitle of your textbook, chapter 4.10, which is the Ramachandran diagram defines the restriction of backbone conformation. But let's expand the sentence a little bit and by asking yourself a set of questions like, what parameter is this diagram using to capture the restriction on the backbone? And more specifically, on a molecular level, what the restriction means. And where does the restriction arise? And since you guys have been talking a lot about the non-covalent interaction between the residues, But notice Ramachandran diagram is really dealing with the backbone, which is covalent bond. So what should we be really looking at? And only after we understand what's going on in the backbone can we understand why the parameter in Ramachandran diagram will be a faithful representation of the restriction on backbone conformation. All right, so let's dive in. This is a Ramachandran diagram. First of all, the process where the restriction arises is protein folding, which involves conformation change. Oh my gosh. Conformational change in backbone, solely arising from the rotation about covalent bond in the backbone, which means it does not involve the breakage or reformation of covalent bond. One exception is the formation of disulfide bridge, where you have two cysteine. You might go through a oxidation and form a disulfide bridge, but this is not necessary for protein folding. Because of the rotation, now we have the torsion, the rotation about the covalent bond. We have the torsion. Is it how it spells? Torsion angles, or dihedral angles, which is the angle between two intersection planes. If you're familiar with OChem, you can understand it from a Newman projections point of view, or you can refer to a textbook and see what are those angles are. So now we have, because of the rotation, we have three angles. The rotation about the peptide bond give us omega. The rotation about the amide alpha carbon give us phi. And the rotation about the carbonyl carbon alpha carbon give us psi. All right, so let's look at omega first. Notice omega is restricted to either 0 or 180 degree because of the planarity of the peptide group, arising from the partial double bond character of a peptide bond. We can understand this partial bond character by a observation of the peptide bond, which is shorter than other bond, and we can infer, and from that we proposed a resonance mechanism that lead us to the conclusion of the partial bond character. And because of the partial bond, it makes the distortion from planarity hugely energetically closety. So that means these four atoms are essentially locked into a plane, which we called amide plane. But I think it might be more useful to think that it's actually a set of 6 atoms fixed into a plane by a peptide bond, so that it leaves the only significant degree of freedom to the torsional rotation of either phi or psi. Yeah. Is there any question about that? Okay, so let's quickly go over, before we talk about finding psi, let's just quickly go over the cis conformation and trans conformation about the omega. Notice the tris and the trans is defined by the position of 2 alpha carbon with respective to peptide bond, and trans is favored because it avoids the collision between 2 adjacent alpha carbon. One exception is proline, which can adapt a cis conformation in favor of introducing a sharp band to the protein structure. But the thing to notice in this statement is not that it introduced a sharp bend, but more about proline. I invite you to think through why it is proline that can adapt a conformation which is disfavored by other amino acid. Okay, so let's talk about phi psi. The two torsional angle defines the backbone conformation. One thing to notice is that this set of phi psi is actually sufficient to describe the backbone conformation because of the uniform stereochemistry of protein backbone, where covalent bound geometry remains approximately constant regardless of what protein the amino acid is in, what position of the sequence the amino acid is in, or the identity of the individual amino acid. So now I think we can talk about Ramachandran diagram, which use the set of phi and psi to describe the backbone conformation by ruling out the combination that introduced a van der Waals portion to the structure. One thing to notice is that you see there's a lot of blank area in the diagram. So this Ramachandran diagram is indeed a van der Waals repulsion diagram, where most phi and psi combination are forbidden. Notice that the original Ramachandran diagram used the hard sphere model for a alanine dipeptide bound, which essentially treat the atom as a hard sphere. So once the collision occurs from rotating the two bound, the combination is deemed as disallowed. So the conclusion is really a binary one where the chance to observe a conformation is either 0 or 1, ignoring all the other stabilization interaction. But this approximation is not a bad one, because van der Waals repulsion is actually so strong that other stabilizing interaction that Ramachandran ignored cannot compensate for this energetic disfavorability. But if you think in terms of protein folding, you can notice something weird, because if the probability is discrete, means it's either 0 or 1, how can this folding actually happen, right? So then a more comprehensive energy diagram, it involves other kinds of interaction, like hydrogen bond interaction with water. It basically provides a softer version of the Ramachandran diagram that it offers the pathway for transition to happen. But to notice that the lowest energy region is still the same as the original diagram. Okay, so we can talk about the allowed region on the Ramachandran diagram. I think this should be fairly just recognizing and memorizing stuff. So very quick, this alpha region corresponds to alpha helix. If value of phi and psi are clustered around -60 and -40. Oh, one thing I really want to notice, because time is not much for me, is this clustered, because you have this repetitive pattern in alpha helix beta strand. So you have this cluster of dots. And beta region corresponds to beta sheets, where the phi and psi is clustered around -120 or -120. And this left-handed region, which is not common, but sometimes it does occur. So in case when you have scattered points, just randomly scattered in these three regions, that might indicate loop, because you don't have this repetitive pattern, but it could be other things. Yeah. So maybe in the last 2 minutes, I want to emphasize this idea of fidelity of the Ramachandran diagram. It's tempting to think maybe the sidechain will make a big difference to the restriction on backbone. And actually, your book give this explanation that because the original one used the alanine dipeptide, so the combination disallowed for the alanine will also disallowed for other bulkier sidechain, which is very true. But also, the influence from the sidechains is actually very small. I have this amino acid Ramachandran diagram corresponding to the amino acid chart, and you can see they're fairly just similar, regardless of the sidechain. And one thing to notice, and you should really notice this, is the glycine, which is achiral because the R group is a hydrogen, and also proline because it has a fused structure. The sidechain fused into backbone, so it has more conformation, no restraint. Yeah. And I have some example of how Ramachandran diagram apply to a real protein structure that you can take a look at. And I have a fun problem for you to think about. And you can this can also help you review the handedness of amino acid. Yeah, I think that is. If you have questions, just put it in the chat and I will take a look at it. All right, sorry for passing the time. Thank you guys for coming.
Okay, so with that, I'm going to